In this segment, we'll understand the C value part. And now we have space so we can write it properly. Now, what exactly is C value? C value is the total DNA content which is present on one set of chromosomes. DNA content on one set of chromosome, and one set of chromosome is represented as N. So in case of haploids, there is one set. Diploid cells, they have two sets of chromosomes, and so on. So this is what is meant by C value, and its unit is picogram. It is always given in picogram. So now let us see how the chromosome number and the C values are maintained. Let us start with this cell, the starting point. It has chromosome number 2n. It is a diploid cell, and one n would have 1c, two n's would have 2c. So at this stage, we will write the chromosome number is 2n and C value is 2c. This cell after G1 enters or comes here by the end of G1. What has happened in G1? Growth, protein, enzyme, ATP, RNA, all those things. But there is no change in the genetic content because DNA replication takes place in S phase. So now the cell is here at this. It has become bigger, so let us draw it slightly bigger. The growth has taken place, the cell is bigger. Chromosome number is still the same because replication of chromosomes or division of chromosomes has not taken place. So here, chromosome number is 2n and DNA content is also same. So C value is again 2C, no change so far. Now this cell goes through S phase where DNA replication is going to take place. And this cell now comes here. This cell has chromosome number. Same because chromosomes have not divided yet. But DNA content has doubled. So in this case here, chromosome number is 2n and C value is 4c. Now how has this happened? For this, we will have to just go over the structure of chromosome and what exactly happens. So if, say this is the chromosome that we are talking of, and we know chromosomes have these kind of chromonemata, these threads which are actually the DNA content which is wrapped around histone, that is the nucleosomes. Now when it makes a copy, when this DNA replicates, then what exactly happens is same thing. This is the chromatid, and now what has happened is this was one chromonemata, this is other one, here also there are two threads, and each thread has a copy of it. So now here we have a copy, here we have a copy, same here and same here. And this chromosome develops a membrane. That means we find two arms which are known as the sister chromatids. But the structure is one chromosome. These are the two arms. So you call this structure as one, but the DNA content has doubled. So here, what is the chromosome number? 1. And its C value is 1. 1 in the sense, whatever the number of chromosome is. Here, chromosome number is still 1. But DNA content is double. The original threads and the copy threads. So in S phase, we still see one chromosome like this. But the chromosome is with two sister chromatids or two arms. So chromosome number remains 1. But what has happened to the DNA content? Whatever was the original, now there is double DNA. So if we talk of one set of chromosome, that is if we are talking of human cells, 23 chromosomes. That is one set. So C value becomes 1C. Here the chromosome number is 2n because we are starting with diploid cell. So normal C value was 2C as we have been writing here. Here the chromosome number is same because unless and until the chromosome splits from here and the spindle fibers, they pull them apart, we will still consider it as one chromosome with double the DNA. So chromosome number is 2N and C value is 4C. Cell, the same cell comes here by the end of G2 phase. Chromosome number, there is no splitting yet. So chromosome number is still 2N and C value is 4C. Now the cell is going to go through the cell division, and as this tells us that it is the nucleus which is going to divide first and then cytoplasm is going to divide. So when nucleus divides, what exactly happens? This type of chromosome, that means the chromosome which has two arms because of this DNA replication. So these two arms, they have their original DNA strand and a copied DNA strand. So here, now this is the original and the copy, and this is the place where the spindle fiber is going to attach. Now this spindle fibers contract, pulling these chromosomes apart, and this is the time when this middle part, that is centromere, splits. When this splits, we get the chromosome number double. So one chromosome is splitting into two. So when the cell divides, that is in anaphase, when these spindle fibers pull the chromosome and the centromere splits, that is the time chromosome number is going to become 4n. Then it divides. 2n goes towards one pole, 2n goes towards the other pole. So here in anaphase, in this stage, if the cell is here, for example, and let me draw this cell here. So if we are drawing this cell, what is happening is, this was like the chromosome was here, and assume this were 46. So 46 chromosomes are going to split. So 46 are going to move here, 46 are going to move towards the other pole, and that is how after cytokinesis we would get two cells with 46 and 46 chromosomes. So in anaphase, the chromosome number is going to change. But here also there is a tricky thing. The cell has not divided yet. So one cell has 2n and 2n. So actually here the chromosome number is 4n and the C value is also 4C. After cytokinesis, that means here in this case, at this situation, when we talk about these two cells, now they have 2n, 2n and they have their normal DNA. So this would have 2C and 2C value. So this is one important thing which we have to remember whenever we are talking of the C value, because chromosome number is going to change only when one chromosome with double DNA content is going to split, and that happens in the cell division part, that is M phase, during anaphase. So chromosome number split and the C value changes when the DNA replication takes place. Now here we will take one simple example to understand how we are going to solve the problems which are associated with this C value. Say a question is asked that a cell, and whenever we are using the word cell and nothing is specified, that means we have to talk about diploid cell. A cell has C value, say 20 picograms. This is the question which they have asked us. What would be the C value in? So we are asked the C value in, say, egg and endosperm. We know that eggs are haploid. So egg is going to be haploid. C value is 20 in a cell. That means we are talking about a diploid cell. So this diploid cell which we have taken here is 2N. So this 20 picogram is actually 2C. If the egg is ours, egg is haploid, so its value is going to be C. That means 10 picograms. An endosperm. Again, when we are talking of simple endosperm, we are again talking about the most common plants, that is angiosperms. And in angiosperms, the endosperm is triploid. So endosperm is 3N. So if it is 3N, N is equal to 10 picogram, 2N is equal to 20 picogram, 3N is equal, will be 30 picograms. So using this, they are going to ask us questions. They may give us directly the cell, or they may just say that the egg of a particular plant has C value 10 picogram. The egg is haploid, so the C value is given is of N chromosome, N number, that is one cell. What will be the number of, or what will be the C value in, say, root tip cell? Root tip cell is diploid. Egg has 10, so root tip will have 2N. Again endosperm. And if they want to complicate this question a little bit, they would ask about a gymnosperm. In gymnosperm, endosperm is haploid. So we'll have to remember, if they have just generally given the question, we'll take angiosperms as the common plants. But if they have specified, like endosperm of Pinus, Pinus is a gymnosperm, or a cycas plant. Cycas is also gymnosperm. So there the endosperm is haploid. So if something is mentioned like this, we'll have to first think whether they are asking about a gymnospermic plant or an angiospermic. And based on this, then we can answer the questions related to C value.